Present our first speaker for this evening, Dr. Daniel Missailidis, PhD. He's a postdoctoral cell biologist and research fellow with the Department of Microbiology, Anatomy and Physiology and Pharmacology from La Trobe University in Victoria. And Dr. Missailidis' research spans multiple cell types and body systems with a focus on identifying mechanisms and biomarkers for myalgic encephalomyelitis to ultimately improve outcomes for those affected. His work places particular emphasis on energy metabolism, and more recently, Dr. Missailidis has explored mitochondrial dynamics and cellular bioenergetics in ME. So kindly welcome to present Dr. Daniel Missailidis. Thank you.
Thank you for the very kind introduction, Stan. Hi, everyone. So thanks for inviting me, and it's my pleasure to give a talk today for Mega. So my name's Daniel. I work on the other side of Australia, down in Melbourne, for those international people that we might have watching this or watching the recording. And I work at La Trobe University, where we have a lab that has been working on ME for quite some time, which I'll get into soon. And we have some absolutely gorgeous research facilities here that I always like to show off to people that haven't been to La Trobe. So I've been working on ME for a really long time now. I started wet lab research on samples from people with ME in 2016, and I've continued all the way through since then, having completed my PhD on ME 2017 to 2021. I currently work in the molecular cell biology lab headed by Dr. Sarah Annesley. We have a few other senior research staff, and here's just two of our expanding group of PhD students, but Tina and Ben have projects that are studying ME as well. So in my PhD, basically what I did was looked at cell lines that we made from the blood of people with ME with a view to see if there was anything going wrong with mitochondria, and identified a pattern of abnormalities there. And from that, we've expanded into basically a whole lab's worth of different research programs. So we've got particularly Wana and Claire looking at the specificity of what I observed in my PhD across different tissues. So the cell lines that I used in my PhD were derived from immune cells. Do the things that I saw there come up in other tissues across the body? Are they specific against other diseases, but especially diseases that might have overlapping symptoms? And how do these changes persist in cells from people with ME? Since I identified a variety of different things going wrong in my PhD, we didn't know what among these things that I identified were the causative differences. So in order to determine that, you have to take, let's say, the 20 things that you've found that were wrong and manipulate them one at a time, turn them on or off, and see if they reproduce the other aspects of the pattern. And so Tina and Ben's PhDs are working on components of that cause-effect determination. And I'll get into later why that's really important for figuring out underlying mechanism and having targets for tests or treatments. And there's a whole bunch of other stuff. I'm not going to go through everything exhaustively, but we've got a pretty varied research program now. We've moved much beyond this cell line work into different tissues and parts of the body, and we're expanding that work. So I'm going to give you a little part of the work today. Everything's published and already available, so I don't want to flood people with data. I just want the talk today to be pretty accessible and hopefully memorable. So the long story short is I work on a lot of different samples from the body, but my particular interest is in where dysfunction in immune cells particularly and potential abnormalities in energy metabolism might intersect in people with ME, and also in studying and representing severely ill patients, which I do by home visits, and I've been doing for about 7 years now. Obviously, people who are severely ill are the people that we need to help the most. And people who are severely ill are also more likely to show differences in the things that we're studying. So it's a win-win for everyone if we can do a better job of including them in research. So as I said, part of my interest is in how cells generate energy. So up here in the left, we got the cute little cartoon of a cell. Our bodies are made up of trillions of cells. And within most of our cells, we have mitochondria, which are these little bean-shaped things here. So if we zoom in, this is what a mitochondrion looks like. And this is where a lot of the energy for our body and for our cells comes from. They're basically the little power plants of our cells. So if we zoom in to the mitochondria, it's got this kind of like double membrane sac-like structure with a space in between the membranes. And so this diagram here is a zoom in on that. So we've got the outer membrane up here and the inner membrane down here, and there's a space between it. And there's a lot of detail that I don't need you and don't want you to understand, but what I want you to take away from this is that there's two membranes and there's these 5 complexes, which are your energy generating machinery in your cell. And these 5 complexes pump protons into that space, and then the protons leave that space through complex 5. That is what makes your energy for your body in the form of a chemical called ATP. So to recap, you've got these little things called mitochondria in every single one of your cell in your body, and it's got these 5 pieces of machinery in it that produce the energy in the form of ATP for your cells and for your body. And this is what a lot of what I look at is based on. So we can measure this by measuring the amount of oxygen consumed at this step by complex 4. We can do this with a technique called a seahorse extracellular flux analyzer. Basically, if we start over here and break it down, these are cells that we'll have taken from patients from various different tissues. Let's just say for argument's sake, as an example, the cell lines that I made from immune cells circulating in blood in my PhD. We grow them up in these flasks, and when we have enough of them, we'll layer them into these little holes in these plastic plates. And at the bottom of those holes, you have a flat surface where you have a single layer of those cells. They're alive and they're intact and they're breathing and eating nutrients. We put a nutrient-rich juice above them, and they'll happily devour that. And while they do so, while they make energy, they'll consume oxygen from in there. And when they consume oxygen, the seahorse machine can actually detect that. So we can measure the rate of respiration. If we go back, that oxygen being consumed here, which is kind of an indirect readout of how much of this whole process is occurring. We can measure how much of that oxygen in here is being consumed by these cells, and it tells you what's going on. So this is an overly complex diagram of all the different things that I've been looking at, but we're not going to go into that today because that's probably hours worth of presentations, certainly too much for someone that has any degree of brain fog. So let's zoom it in a little bit to our 5 complexes again. And when we look at these 5 complexes using this seahorse technique, what we've found on average in about 160 people that we've looked at now, there's an elevated capacity for energy generation by those 5 complexes, which we call respiration. But complex 5, which is the one producing that ATP, which is the chemical energy of your cell, is doing less of the work proportionately, which is really unusual. And what it suggests is that the cells are doing other things with the mitochondria. Now, if I go back to the complicated diagram, you don't need to worry about the details, but what you can see is there are other things that span those 2 membranes, and they can also consume these little guys, the protons, that are pumped into this space by respiration. So for whatever reason in our cells from people with ME, a lower proportion of this is going through our complex 5 and making ATP than in a healthy cell, and more of it seems to be going towards the other processes. So there's a lot of possibilities for what this means in terms of pathology, but one example I'll give is that if we look at the levels of those complexes, they're increased in the cell lines from people with ME on average, which explains that elevated capacity. And if we correlate that against orthostatic intolerance, which is the main reliable bit of clinical data that we've got across all of our different cohorts that we've been recruiting from different sources over the decade, if we actually correlate that, as the capacity for this respiration goes up, the severity of orthostatic intolerance goes down. How this is happening, what the mechanism is there, or what the association is, we don't actually know. That's something we've got to look into, but it suggests that whatever might be happening in these cells has some degree of relevance to the patient. So in terms of relevance to the patient, obviously everyone's favorite word is biomarker, because we need blood tests for the disease. I'm not going to come out here and say we've got a blood test, because we're nowhere near that, but just pointing out that if you take some of the measurements that we've done over the years, so if we combine them here, there's about 7 different parameters there, and you produce a little plot here with CFS up here, the ME, sorry, up here, and the healthy controls down here. You see a decent degree of separation, and in these plots here, the more of this is grey, the greater the accuracy of the discrimination. So if you use these things to separate ME samples and control samples, the more full this is with grey, the more accurate the test is. And I found a combination of variables in my PhD that, as you can see, completely separated control and ME samples and were accurate at discriminating them. But this was all done in small cohorts with really highly specialized methods. So what it tells us is things that we see going wrong in cells with people with ME can separate them from healthy people and have clinical relevance. But in terms of a blood test, there's a few challenges in the way, and those challenges are we need to validate it in more people, and we need to validate it against symptoms that have against diseases that have overlapping symptoms, or syndromes that have overlapping symptoms, because those are the things that would be likely to confuse diagnosis, and also to translate this across to measurements that are more clinically accessible. Since a lot of this is really expert stuff that takes literally months to do with people that have been trained for years doing it, it's not viable for a blood test. So we're working on getting these kind of practical elements worked out at the moment. But ultimately, to reach a diagnostic test, we need to pin down mechanism. Once we understand the specific things going wrong in people with ME, we'll know exactly what to hone in on and what will be specific versus other diseases. So mechanism is really the key in searching for biomarkers, as it is as well for treatment targets, of course. So we've looked at other tissues, as I alluded to earlier. We've got a cohort of about 50 people using skin biopsies, which we can turn into cell lines called fibroblasts. And these are these cells here. They look really cool and they're really fun to image. But what we found in looking at these energy pathways and mitochondrial function in these cells is that there's a different pattern of differences compared with the LCLs. LCLs are the immune cells that I've been talking about earlier in the talk. So what this suggests to us is that those respiratory changes that I was guiding you through earlier seem to be specific to immune cells, or at least that immune cell subtype that I was showing you. And in many of these cases, we've actually taken fibroblasts and lymphoblasts, the LCLs, from the same person with ME or the same healthy control. So we've got a really tight frame of reference there, and we still see that difference between tissues. So we've done a few little exploratory things that might be interesting for you all to see tonight using those fibroblast cell lines. We've got some collaborators next door who are experts in a little thing in the cell called a lipid droplet. What a lipid droplet is it's a little fat storage compartment in the cell that's important for metabolizing fats and in promoting immunity, particularly against viruses. And in a small sample, we found that the number of lipid droplets per cell in the ME cells was reduced and their size was increased. We obviously don't know if this means anything mechanistically, but it's one of the little exploratory pilot studies that we've done recently, and it's generated us some pretty cool pictures. It's kind of hard to see that difference here, but if you quantify all these little green dots, which are the lipid droplets, you would find that there are fewer of them but larger down here in our ME samples as compared with our healthy individuals. So the current study, the most recently funded study that I'm doing, is one where we're trying to integrate all that we know about ME from our work and from other people's work in a kind of multi-system sort of integration. So we've sampled 100 people. All the samples are done and we're starting experiments soon. The idea is that we're going to do really deep immune profiling of many different aspects of the immune system. We're going to look at many body-wide indicators of energy provision and metabolism, as well as deep characterization of gut microorganisms, including classes of organisms that I don't think have been looked at yet, as well as levels of particular nutrients in the gut. We're going to look for not only signatures amongst these different domains that correlate with symptoms in people with ME, but also associations with each other to potentially establish causal pathways that we can look at later. And then along the way, we're going to keep an eye out for druggable mechanisms with a machine learning screen of all the tens of thousands of different things that we're going to measure. So it's a really cool study, but we'll see where it goes in the next couple of years. And this study is actually a good example of why we need to continue to and increase how much we involve health consumers, so being patients or clinicians, particularly patients, in research. Obviously, it kind of ensures the accountability and transparency of your research and sensitivity and inclusivity of it. But really importantly, it's making sure that what you're doing is actually relevant to the people you're trying to help. And if you have people that are sufficiently informed, which is who you'd hope you'd be choosing for these kind of things, they'll actually help you refine the study design. The more people that you have looking at something that know what's going on, the better, really. So examples of changes that we made for this study that I was just talking about, we switched from planning to use Canadian 2003 CCC to using international consensus criteria with a view to make sure that we capture severe patients, as is one of the priorities anyway. So that was a really important change that was actually encouraged by a patient. And also to introduce formal risk management plans for risks to people with ME, such as what happens if someone has a really bad PEM flare-up afterwards. How do we help them with that? How do we help avoid that during how we communicate with people and how we deal with them throughout the study and things like that. So just to take home from this, engaging informed stakeholders throughout study design and execution is valuable and ethical, and I highly recommend that all researchers do so. So if there's one thing I want you to remember from my talk today, it's that ME involves diverse changes to immune cell metabolism. I showed you just one or two of those things. There's been many studies around the world in different types of cells with different types of techniques, looking at different aspects of energy metabolism, and something always comes up as being abnormal. So the source of it and the relevance of it in terms of consequences on the whole person, that's what we've got to figure out. But the signs are there. In terms of specific meaningful outcomes for patients, which is what we all want ultimately, particularly drugs and disease-specific biomarkers, there's two things as a field that we really have to determine. The sequence of what's going wrong, so cause versus effect. So let's say in whatever a particular lab has looked at, they found a few dozen things that stand out as being wrong or whatever that might be. Like I was alluding to earlier with Ben and Tina's PhD projects, manipulating those things one at a time to see which problems lead to the other problems will help you establish like a causal chain of events. If you work back up the chain to the initiating things, you have a better candidate for a drug intervention. And then particularly that link from what's going wrong at the cellular level to symptoms is really important. You have to kind of rationalize that when you build a disease model, and that's introduces other potential intervention points. So there are other immunological conditions where that whole chain of events that explains the clinical picture is known, but just drugging that upstream initial driver is not actually enough. You might have 3 or 4 different links on the chain that lead to different symptoms that get targeted concurrently. So we kind of have to build that into our models as we work on this stuff. So I get asked often, can we expand our research to recruit interstate? And I just thought I'd update everyone and say yes. I've been working with companies to make this possible, and for future studies, we'll definitely be doing it where we can afford to do it. If you're interested in participating in our research, you can just Google me, and you'll find my email address on my website over here on the left-hand side. And if you're interested or have the capacity to read the papers, they'll also be in the research outputs tab. If you're in Victoria, Queensland, or WA, there's long COVID research relating to pretty Australia-wide biobank and COVID-19 studies run by the GRA lab at La Trobe Uni, which are an immunology lab. You can register for that study using this QR code. So if you're interested, just take a photo of that and that'll lead you through to the registration form. And we'd really appreciate anyone who's interested in COVID-19 or long COVID research getting involved that way. So I was asked to kind of give some aspects of my journey relating to positives, maybe low points and hopes and wishes. So as far as my experience goes, the hardest thing about researching ME is the big elephant in the room, which is the funding. Keeping the research going is tough. The fact that we've done it for this long is a miracle, and it relies largely on philanthropy a lot of the time. The biggest positive for me recently, I've spent the last probably 7 months going again to meet a few dozen people with ME at home, and just talking to people with ME who are really severely ill, and them finding out that there is research happening, in Australia, is really making a difference, because it's giving people that assurance that there is progress happening. And in terms of hopes and wishes, there's really only one. That's the one thing we all want. Everyone with ME gets better. I'm not going to belabor that or overcomplicate it. We want everyone with ME to get better. And I think everyone researching the disease would be happy to be out of a job looking for research jobs in other fields because we fix ME. So that's what we're all aiming for. So I just want to acknowledge there's a lot of people that were involved in even just the tiny, tiny little snippet of stuff that I gave you today. Too many to acknowledge in detail, but particularly the people from our lab who taught me everything that I kind of learned and knew throughout my PhD and now bring as a postdoctoral scientist to the field. And participants and carers without which we couldn't do any of this, and particularly the funders as well, who enable the research. I think that was my last slide. Yep. So thank you for listening, and I'm really happy to take any questions that anyone has.
Great. Thank you so much, Daniel, for sharing your work. It was good to see that it was nicely summarized and digestible, which I think can be quite challenging to do for such an extensive research career. Good. Does anyone have any questions to kick off? Otherwise, I've got a few. All right. I'll start off. So, what really fascinates me about ME is that it's not a straightforward condition. We see a lot of variability in the clinical presentation and what triggers the initial condition to manifest in people. So I was wondering whether the data is available now, or maybe it's a future direction for you and your team. Is there an association with how complex 5 works in the generation of ATP, in particular subgroups of ME clinical manifestations, or are you seeing it's relatively uniform that this complex 5 impairment is present in ME?
I don't think this is going to be like a uniform thing necessarily, because the only thing I would have confidence saying that for would be if we identified like some core underlying pathology that caused the disease for everyone that has it. The reality is, as we know, there may not be one underlying mechanism, and we certainly haven't proven that this one change is the underlying mechanism. It's just one thing that we've seen different as everyone does in the research. Now, in terms of relationship with particular clinical elements, we've sourced patients from different clinical sources over the years. So we have different combinations of clinical data, but across that larger cohort, the thing that we do have for everyone is the orthostatic intolerance, and it does correlate the severity of that, not impairment, but reduction in proportional workload being done by complex 5 does correlate with the orthostatic intolerance. So there's definitely something there. In terms of how universal it is as a mechanism, I'm not going to say because I can't say because we haven't demonstrated that. But it's, as always in ME, with all the research, the findings are as variable as the clinical presentation is. So I think that what you were alluding to with subgroups is a really important thing that we have to move towards. And the current study that I'm doing at the moment has been designed from the ground up to capture that so that we can take that wealth of data that we're getting, but also apply it to the relevance of that variable clinical picture.
Exciting. So watch this space. Good. So it sounds like the goal will be, yeah, like every research group is to extend the breadth of that investigation and make it more applicable to more people. Yeah, great. Any other questions? I'll read this one out from Jenny. So your research relies largely on philanthropy for funding. How ME community help to access government, or how can the ME community help to access government funding?
Hi, Jenny. Thanks for coming along. It's good to see you in the audience. That's a very good question and not an easy one to answer. So if we think back to the most success the field has had in getting government support pre-COVID, that would have been the targeted call for research, obviously, in 2019 or whenever that was. And that was based on, I don't even want to know how many years worth of lobbying. So I guess there's two elements here. There's the pre-COVID challenge, which is needing that amount of lobbying, which needs grassroots action, but also nobody can realistically ask that much work of people that are sick. So I guess keeping the advocacy alive is the simplest thing I can say there. In terms of the landscape being different now because of COVID, there's obviously a lot of money in the system at the federal government level, specifically going to long COVID. In order to fund ME by that, there has to be acknowledgement of the overlap, that Venn diagram, long COVID, ME, and the people that have PEM and satisfy ME that got it from COVID. That overlap needs to be more well known, I think is probably our best route to mobilizing that money that's already there. And government is receptive to stakeholder voices in changing how that gets deployed, at least from what I've seen in things like MRFFs. So I think spreading awareness of the relevance of what we're doing for long COVID to ME is the best strategy we can do. And I think in terms of any kind of change making or advocacy, having that one unified point that everyone's hammering home is the effective thing. And so I'd say that would be the thing to focus on. There's no easy answer.
Right, thank you. So it sounds very much, again, that, yeah, just keeping advocacy alive, keeping ME in the public spotlight in events like these and supporting work and disseminating it to the public is really important. So I guess everyone here tonight is part of making that achievable. We've got time for maybe one last question, if anyone has one. I have one, but I'm happy to withhold it if someone else has something more interesting. Okay, my other question, Daniel, is I was wondering with the um, mitochondrial changes that you and your team have identified to date so far, is there a theory as to how these may manifest in the first place? If we've got some sort of trigger damaging this system, is it like a change in expression or post-translational modification or something like that? Or maybe another mechanism that I'm not aware of? What are your thoughts?
Yeah, so obviously there's nothing clear in terms of genetics that's come out. So I don't think it's a genetic thing, considering that what we're seeing seems to be isolated to B cells, because well, at least that's what we've looked at. We've only seen evidence of this in cell lines derived from B cells. It's likely to be an immune isolated thing. Jury's not out on that, but that's the likelihood at the moment. So in my view, the most likely simple explanation is that there's either an aberrant signal to the immune system that's leading to downstream cellular changes, or something different about, say, a particular receptor that's oversensitizing a particular immune cell subtype to that signal. And so when that signal comes along, which might be invoked by, exertion, the immune system freaks out and then leads to all the downstream changes that lead to pathology. Obviously, that's not based on experiments we've done, and no one has figured this out, but a lot of people are thinking this, and in terms of my view from our evidence and our research, that's what I think is going on. It is possible that an underlying defect in energy production is present in those immune cells, and then when that signal comes along, they respond to it abnormally because the underlying metabolism is dysfunctional. That's also possible. I just think an explanation for that is a bit harder to find in the absence of a genetic cause. It may be that there's a more subtle combination of things that lead to a change in homeostasis that then causes this. But we're looking at things outside the body too here, so that's tricky.
Yeah, thank you. It's not a simple thing to accomplish, discovering the answer for what's actually causing ME. So that's, yeah, thank Daniel for presenting on his extensive work for ME and mitochondrial metabolism in this space.